Okay, let's just dive right in. For centuries, right, we've studied the building blocks of life using microscopes and lab experiments. But today, a brand new, powerful collaborator is stepping into the lab, artificial intelligence. We're going to explore how this technology is becoming this incredible molecular problem solver and how it's literally changing the way we do science.
And this isn't some far-off future thing. It's already happening in hospitals. AI is being trained to automatically look at medical images, like this chest X-ray here. It can learn to spot these tiny little abnormalities or patterns of disease, leading to diagnoses that are not just faster, but potentially way more accurate than what the human eye can catch on its own.
But it goes so much deeper than that. I mean, all the way down to the molecular level. AI is now tackling one of the biggest challenges in all of biology, predicting the complex 3D shapes of proteins. This is a puzzle with more possible combinations than there are atoms in the universe, and understanding these shapes is absolutely fundamental to figuring out how diseases work, and more importantly, how we can design new drugs to fight them.
So yeah, this is not just one or two cool applications. We are talking about a full-blown scientific revolution. From analyzing our genomes to developing what's called precision medicine, treatments that are tailored specifically to your unique genetic makeup, AI is quickly becoming a tool we just can't do without.
Okay, so this all sounds amazing, but it brings up a really important question. How? How does it actually work? What do we even mean when we say that a machine learns something? It's not magic, I promise. It's a very specific and pretty fascinating process.
At the very heart of this revolution is a concept called machine learning, or ML for short. And the core idea is really this. We're teaching computers to learn directly from data. That way, they can make predictions or solve problems without a person having to sit down and write out explicit step-by-step instructions for every single possible scenario.
The source material gives a brilliant analogy for this. Imagine you're teaching a child to tell the difference between healthy cells and cancerous ones. You wouldn't write out a long, boring list of rules for them to memorize. No way. You'd show them hundreds of examples, right? Pointing out the features. And eventually, they just get it. They learn to recognize the patterns all on their own. Machine learning works in a very, very similar way. And this visual really shows you what a complete flip in logic this is. With traditional programming, over on the left, a human writes the rules. You feed in data, the rules are applied, and you get answers out. Simple. But with machine learning, on the right, you flip the script. You provide the data and the answers, like, these images are cancer. And you make the machine figure out the rules for itself. It actually learns the how.
So, how do scientists actually do this? It's not about just hitting a big learn button. It's a structured, rigorous workflow, a lot like any other scientific experiment. So, let's walk through it.
This lays out the entire 6-step journey. And it all starts with a solid study design, because you can't get a good answer without asking a really great question. Next come data collection and preparation. And let me tell you, this is often the hardest part. Getting clean, high-quality data is everything. Step 4 is model training. This is where the magic happens, where the machine actually learns. But you can't just trust it blindly. So step 5 is model evaluation, where you rigorously test its performance. And finally, step 6 is interpretation, where that human expertise is so vital to make sense of the results and uncover real scientific insight. Now, not all machine learning is the same. There are kind of 3 main flavors. Supervised learning is what we talked about with the cell example. You have labeled data. Unsupervised learning is when you don't have labels and you just want the machine to find any hidden structures or groups in the data. And then there's reinforcement learning, which is all about an agent learning through trial and error to achieve a goal. Let's take a closer look at the first 2, since they're the most common in biology.
And here's what that looks like in practice. On the left, you've got supervised learning. The machine is given labeled data. It's told which dots belong to which group, and its job is to learn the boundary between them. But on the right, with unsupervised learning, it just gets a jumble of unlabeled dots. Its job is to discover the underlying patterns or clusters on its own.
Okay, so, as with any powerful new tool, machine learning comes with its own set of challenges and potential pitfalls. It is not a silver bullet, and using it responsibly is absolutely 100% critical.
I love this quote from Alice in Wonderland because it perfectly captures the most important first step in any ML project. If you don't have a clearly defined goal, if you don't know where you want to get to, then even the most powerful model in the world isn't going to help you. That initial study design, well, it's everything.
And there are some real technical hurdles to watch out for.
For instance, a model can overfit. It basically just memorizes the data you showed it instead of learning the general principle. That means it's going to fail on any new data it hasn't seen before. The flip side is underfitting, where the model is too simple to capture the real patterns. Then there's the famous black box problem. Some models give you amazingly accurate answers, but you have no idea how they decided. That really undermines scientific trust. And finally, the most critical rule of all, biased data in means biased results out, period.
And beyond the technical stuff, using these tools in the real world means we have to wrestle with some really complex practical questions. Things like, is the model fair? Does it work for everyone? When we're using sensitive patient data, is that data protected? And who's accountable if the model gets it wrong? There's a constant push for greater transparency so we can actually understand and trust the models we're starting to rely on. So, with all these complexities, what is the right way to think about AI in science? Well, the key message from our sources is this. This is not about replacing human scientists. It's about empowering them.
Here's the bottom line. Machine learning is a powerful tool to augment and enhance human expertise. That deep knowledge of a biologist or a clinician is more valuable now than ever because they're the ones who can ask the right questions, prepare the right data, and, most importantly, interpret the results in a way that's actually meaningful.
And for any scientists out there thinking about getting started, the path is actually pretty clear. It begins with learning the core concepts. You don't have to become a master coder. It thrives on collaboration between the science experts and the ML experts. It relies on scientists knowing their data inside and out and using that expertise to pick the right problems for this powerful new tool to tackle.
So, machine learning is no longer some futuristic concept from a movie. It's here, right now, acting as a new kind of collaborator in the lab. And that leaves all of us with one really thrilling question. With this powerful new partner at our side, what great mysteries will we solve next?